Hi everyone. In this video, we will explore CASIA, the collaborative agent system for single cell interpretable annotation. This is another recent tool which is AI based, LLM based, and it also helps in automating the annotation. It is very much similar to what we had discussed earlier. Another tool like MLLMCellType, where it uses multiple LLM to give you a consensus, and and also there is a consensus checker is there. And previously we talked about CellLlama, which is single model based or uh, uh, um, annotation tool. And the advantage was like it was supporting Ollama model, but the previous one was not supporting edge of now. The local LLM model, you have to use the public this one tools. But here the beauty is like you can use a local LLM model for free. Okay, so that's the one pros I would say. So how does it work, this tool? So this is the paper. I'll just show you. So basically, users input, you need to provide what species it is, okay, and what tissue it belongs, and you just simply provide the marker list, and you can provide the different LLM models, okay. And basically it does the 5 stages. So there is an annotator is there which will annotate, then there is a validator which will validate, and then it will format, and also optionally it goes to the scoring as well, and then formatter, scoring, and then reporter, and then it will give some output like what is the general type of annotation, subtype, other things it will do. Okay, apart from that, it also supports some more advanced complex, like some, if you have some RAG database, where you have some domain information about the cell, let's some selected publications are there, you can put it into a database, and that is also the annotator will probably use the RAG. Okay, and it also provides some additional functionality like annotation boost, uncertainty quantification, and subclustering. So basically they have different flavors. If you can go to the website, you will get in more idea. So yeah, okay, this is the paper, right? So yeah, this is the paper. And so let me go back. So this was the paper published in 2026, very recently published, and this talks about, right, the multi-agent large language model for automated and interpretable set annotation. Okay, and yeah, so that's what they are saying. These different stages are there, and then it does. Okay, but let me come to show you the website. So this is the website, casia.bio. You can just give it a quick try. So first we need to import our API key, and so here you can update it. So I have already uploaded my Open Router API key, and Open Router is as I said in the previous this one. Okay, and so the Open Router, right? So this is a unified interface where you just pay to Open Router, and and you will be able to use connect to multiple other provider, like ChatGPT, Anthropic, everything. So you don't have to have separate API keys for everything. Just one Open Router, you can decide the model. Okay, so I for this thing, right, you can use that one, or you can also configure individual as well. Okay, so once you do that, right, so step number one, you have to configure the API key, and you can validate it. Then you can upload your find all marker, that CSV and Excel file you can upload, and you can select the module and then run and save. Basically, this is the batch mode, is what for single cluster it is important. So you should start with this batch mode. And if you have, let's say, 10 clusters, because for example, Surat output will have, let's say, 10 clusters, right? That if you give, it will process each of them in parallelly, and also it will multiple marker ranking method supported, and it will automatically convert gene ID to gene symbol, and HTML report and everything. Okay, so the second approach is CASIA pipeline. This is like even important, like advanced one little bit. So for example, here annotation will you can do furthermore quality scoring and validation. This whatever the output we get validation, and sometimes also it supports something, and I don't know what this merging and grouping, but yeah, it's something that's what boosting it supports challenging cluster. Like for example, what I understand from this, like if there is a cluster where confidence is low, we can further boost analysis for that. Okay, so to start with, right, you can go with this one on UI. So let me do that, but yeah, I'll not do here, but just giving you, like I have configured my Open Router. You can all parallelly do other as well, and yeah, You can test your API as well, and now you can choose the model. Okay, so for example, for best, so you can also choose for the performance selected, automatically selected. Yeah, so you can select what model you want. Okay, this is for the performance. Okay, if you want balanced, means like, low cost, fast speed but medium, so you can go with Gemini 3 Flash, like that, but for that you will have to use that from open router, it will use that model, and you have to pay like that. And yep, so you can select what model you want, and yes, you can upload the this one. So if you let me show the format. See, if you see, the format is very much similar to this Surat output, right? So we can just simply download that CSV file, and you can upload, or alternately you can simply say cluster 0, these are the markers, cluster 1, these are the markers, and it will you can run it. Okay, so this is how it will look like, but I will show you the alternate approach, which is the free one, where we will use programmatically and do it locally, using Ollama model. Okay, so that's about this one, and yeah, so this is what further documentation they have given. Okay, and that you can go R documentation and Python documentation, and the main is this GitHub repository, the CASIA GitHub repository, and that's see, like it is support for R and Python both, but the challenge is like everything is mainly Python based, but what they have used, like, they are using reticulate in the kind of, running Python program inside R. Okay, so that's why it is little unstable, I would say. I tried to when I tried to run R version, when I was trying, somehow my system, it was giving some error. I could not I did not spend more time to fix that. Okay, but it was very challenging because it was asking me to install R tools 4.5, 4.4, so many combinations for that. It was very giving me headache to install this. So then I thought Python is much simpler, because it's Python native Python based, so you can directly go here, and you can follow the the methodology here. Okay, but now, as I said, you can provide different models here, and different API key you have to set. Okay, so you can simply import CASIA. You have to install it first. Pip install CASIA will install it, and then you import it, and you can set an API key, and you can say and give the key and what provider. Let's say Open Router, or OpenAI, Entropic, or DeepSeek, like that. Different provider you can provide. But for local LLM, the method is very simple. Okay, I'll just show you here. Okay, so here in the script, what I have done, like I am importing CASA, and I'm just loading Panda here, and so basically this is how I have set up my Ollama key. Okay, so this is the API, because local Ollama, right? Okay, just to, if you are new to this one, just I'm telling, like Ollama is the alternate API provider which you can install locally, and it will run. Okay, so let it run. Yeah, so you can directly install by this, and then you can pull a model, like Ollama pull a given model. So in my system already, I will just show you. Okay, Ollama list, if I do, right, so in my system I have already installed this powerful model GPT OSS, along with this Llama 3.2, which is the smallest model, a small model, which is it will run in your laptop. Okay, so that's what you have to specify. The key, any key is fine here. Dummy is just any key name, because it's locally running, right? It's not an actual key, but this URL is the endpoint where Ollama server is running. So when Python will connect and submit to this LLM model. Okay, so that's what the first, you have to just set the API key. And so here in this, I'm loading my marker file. So this contains all the marker file, and yeah, so, and then I'm just selecting, because just to demonstration purpose, I'm just selecting cluster number 1, so that only those markers we will try to annotate. And this is the simple command. Okay, first I will not go to the pipeline mode. I will go with the batch mode, which is, as I said, the first analysis, right? So this one, simplest one. Okay, pipeline is little complex because scoring, and so it is multiple stage. So first, if you want to try, go with the batch mode first and see if it is working, then you can move to more advanced one. Okay, so here you say the what marker you want, and the folder name where, and this is the provider. So here you can give local Ollama, or alternately you can give OpenAI this thing, and the choose, I am using model this one, and the tissue is brain and species. Okay, so that's all, very minimal script, this much only the command. Okay, so now let me run three main dot, sorry, run CASIA.py. Okay, it will take some time, but I will show you my previous result meanwhile. So I was getting some errors, forget that. So here, from here it is starting. So first what it happened, like it came here as per the code. So loaded, so it says, how many marker genes loaded here. So total 200 marker genes across 11 clusters. This is my top marker file from the pseudo what I have been generating, and I'm just showing first few rows. You can see that these are the cluster 0 marker. Okay, this is the marker for the cluster 0, and these are the gene, and like that. Okay, and so yeah, so next it says running CASIA annotation with Ollama model. Okay, here, because what I have given here, the model is 3.2. okay, at the, I think that time 3.1.1, why it is using that. Okay, I have to check this one, why it is printing 3.18b, but because I have provided 3.2, right? So yep, but it should run that. So then what it will say, it will select top 20 genes. Okay, you can set the parameter as well, how many genes you want, and then it will say that all the analysis are completed. Okay, so not much log here, so it's now running locally. So it will run the output in my here. So okay, it will give me output like results conversation, result report, and result summary. These three files it will give. So let me show you the output. Okay, so okay, yeah, this is my local output. So this will the output of this one will look like the result report HTML file. So this is what you get, right? Once it is finished, right, you will get these files. Okay, so all the analysis have been completed, and the results is stored over this one, and some additional file it gives. Okay, so let's go to the report. How it will says that when it was run, total clusters was 1, and that's why I have given only cluster 1, right? If we had have given the entire data, it would have done the 12 cluster analysis. Okay, and yes, so it took 20 marker, and it did one iteration, and the final subtypes like CD4, CD8, T follicular cell it could detect. And if you just click on the view full analysis, it will see, it will give you completely. So this is how it is saying, like, I will follow the steps to annotate, list the functional, and all this is from coming from the information from the analysis that LLM did. Okay, and it also tried to cross reference some databases like singlecellatlas.org, and it also determined the most probable general cell type. Then it identified the top 3 most probable cell type, and then at the step number 6 it provided a concise summary of your analysis. So then after that, final annotation completion, then it did the validation. Validation is passed. So then what valid, that's why I said the 5 stages, right? In the stage number, so annotation is done, then it comes to validation. So validation is now another, a second model. It is again try to validate that the provided annotation results appear to be well structured and clearly explains the reasoning behind each step. Okay, the markers are consistent with respect to, like that. So that's why again this validator is now suggesting, like, say, hey, I would like to suggest few minor improvements. So it's telling to the other agent that marker consistency, mixed cell type consideration, and subtile annotation. Okay, but overall it annotation to be accurate, but minor adjustment to, like that. Okay, then after that, the third step is like formatter. Okay, so it comes to formatter, and it is giving the final output over here. Okay, since the next step is the scoring, and reporter is not part of the this one, that's the part of the pipeline. So when you are running this pipeline mode, right, so it will further do the scoring, and then it will generate a final consensus report, and it will also do some boosting, and also lot many things are covered in the pipeline stage. Okay, this is how you will get it. And if you just run this one, right, this will, it gives some summary file as well. Oh, already I have opened. So this is the summary file. Cluster 1 predicted general cell type is T cell. Detailed one is this helper one. Okay, and these are the mixed cell type, and these are the list, marker list, and what model, what tissue, like that. Okay, this is what one summary file it will give. And yeah, this is the, I think this is a prompt template, how it will follow. I think that is the one. So first annotation, right? I will follow the steps. So let me just word mark. So this is how the annotation step, the prompt it went. Okay, so I will follow the steps to annotate this one, and step one, this one, step two, step three, like that. It goes, and validation, and then formatting, and see scoring is empty, right? That's what I said, like the scoring did not happen in this mode. Okay, and okay, so but you get an idea, like it's very simple to do on UI, but programmatically also just one line of code, this much only to do this, the run CASIA as a batch mode. Okay, alternately we have something called yeah, this is the, you can go over the repository examples Python, you can see. See here, you can import the API key, and you can get some marker list here, and this is the only thing, fast mode, right? So that means here you are running the pipeline mode. In the pipeline mode, you have to give again the tissue, which species it is, and what is the marker, and how many thread you want. And now this is the difference what you see. So here what we are seeing, one is the annotator and one is the scorer, right? So here annotator is there, that will annotate using some model, and scoring is done by another LLM model. So here you can choose different model. So that's what you says, okay, who is the annotator provider, let's say open router, and annotator model is maybe some Gemini, whereas you want to score again some other thing from other models. So like that also we need to provide which are the annotator model, which are the scoring model. And if you want to use boosting as well, so then you can also further provide the boosting model for boosting stage, what model and what provider you want. Okay, so that way you can also run the pipeline mode. But batch mode is very simple, as I said. You can provide the command like that, and furthermore you can also do quality scoring. That means whatever the output you got, you want to just score it, so this stage alone you want to do it. So that way you can also run that stage as well. Then some other analysis, I have not tried it, but it's possible. So but see, it's very implementation is very easy. Okay, because they have made a very nice wrapper. You just have to pass the input and choose the model, and then you can play around it. It's so much simple, but only thing, like the cost is here, because and you will it's all about LLM based, right? So but yeah, so ultimately the main summary is like the annotator, validator, formatting, scoring, reporter, and further mode you can enable RAG as well, and you can use some databases to plug in here in the RAG, like cell marker, anything to more extra information you can get. Okay, so yeah, so that's all I had to show in this video, and definitely you can go with the documentation to know further here, but you got an idea, like you can try with the UI one, and you can try with the programmatical one as well. And whenever you want to get some time, when you get some time, you can read the paper to just understand, like how they have done the benchmarking and how the report will look like. So very nicely done, and very latest paper, 2026 paper. And I think again, I'm telling, this is all LLM based. That means they are trained based on the text information what they have. But still, you need to combine the three approach, like whenever possible, the marker based, the reference based, and the AI based together. You can do in confidence, very confidently you can do the annotation process. That will conclude my series here, because yeah. So we have started with I can quickly just show you like what all we covered. Like we started with annotation, very basic, why it needed. Then we covered the marker based annotation, started with that, and we first explored different databases for marker based, like CellMarker, PangLawDB, AgMuth, MSigDB, those kind of thing we explored. And then if in case of marker, we talked two different approach. One is we started with our own method, where I showed like how you can manually do overlap. Then we explored these three tools, different method, like cluster mole, which in a single command, it will compare against 7 database. Then we talked about SC type, where both positive and negative markers are used. And another third type is like CellType, more of an enrichment kind of method, using which this marker based method work. Then we move to reference based, where we talked about single R and cell typist. Unfortunately, I could not demonstrate the azimuth because it's still something I was struggling with the installation. But you can try that azimuth one. The currently at this stage the online app is not working, but you can try the azimuth tool as well. I did not cover that. And then we move to the AI assisted annotation, like we three different approaches I showed. The first one started with CellLlama, where one model, one local LLM, and then it was very, this is purely LLM based, okay, where your it's not where your gene expression, I mean the information is converted into a text, and that text is fed into the model, okay, embedding model, and it does that. Then we moved to the multi model kind of thing. So these two are very similar, MLLMCellType and CASIA, both are same type, both are using multiple agent to do the consensus based analysis. Like again, they have the annotator, validator, those kind of concepts are there. So in short, my suggestion would be like, this is a very complex topic, right? And so four things you need to know, like I said, that means this everything you have to use, like the you can leverage the database information, you can leverage these tools from marker and reference, and then take the AI assistant help. Together you can do a very nice annotation method. And last and final thing I would conclude, like you can use, for example, cloud, that's the one of the good tool, where you can document everything in a skill document, okay, and where you can guide that agent, right? So you can guide this LLM model, so that, hey, see, this is the databases, you can use this tool first to generate this one, and then you can also use these tools to do the reference based, and then you can use this to AI assisted, and then give me the consensus as well. So that means you can use a skill document and guide your method to or guide your LLM, what do you say, AI agent, to do this analysis for you. Okay, so I have created a separate video series. I've been talking this again, like where like how to use cloud desktop, right? Cloud desktop or a cloud desktop to do the single cell analysis. So you can check that, and the same thing you can very easily incorporate in your research to do that. Okay, in short, see, my idea was to just give you a complete like overview about what are the different things are available, and there are n number of tools are there I could not cover, and it's not possible to cover as well, but everything will fall under this three category. And yeah, so don't trust AI blindly, don't like their but always these two are the also a gold standard. Still combine this all three to come to an conclusion. Okay, I hope this series was useful. It was very hard work for me to go through all this gather all this information and collect in the form of a series, and I did my best, and I took a help from so many places, and I could not acknowledge, but everything is available, and the original authors, the documentation, the tutorials, what they provided is amazing. And that's what like this field, and yes, I'm always there to improve. So if please do provide feedback, uh, and it will help me to like understand like where I need to improve. At the same time, if you feel that this content is very useful, and your friends, everyone need, so please do shout out and share this among your network, and yeah, that will be very helpful. So with that, I will stop here, and maybe I'll create some new other series in maybe genomics, and something I'm thinking down the line. So yeah, I will see you in the next series. Thanks for your time. Thank you so much. Bye.